Hey guys, Carlson here to move on to Chapter 9 with you over the general and special senses. We're going to start off with Section 1, and right away you're going to notice that you hear terms related to our Chapter 8 nervous system. So you should find a little bit of this slight review, but we're going to get into more depth about our sensory receptors and how they connect to our internal and external environment. All sensory information is picked up by our sensory receptors, and these are specialized cells or cell processes that monitor conditions inside or outside the body. Our simplest receptors are the dendrites of sensory neurons that we're going to call free nerve endings. They're sensitive to many types of stimuli. Other receptors are sensitive to only one type of stimulus, and the actual area that can be monitored by a single receptor cell is called the receptive field, and that's shown in the diagram here. So we have our free nerve endings of this sensory receptor, and you can see the receptive field that it can actually receive stimulus from. Now, the larger the receptive field, the poorer is your ability to localize a stimulus. So locations like your tongue and fingertips, they have receptive fields that are less than a millimeter in diameter, which can be more precise. All information arrives at the central nervous system in the form of action potentials, which we actually call a sensation in this context. And the stronger the stimulus, the higher the frequency you're actually feeling that stimuli. The routing of sensations is according to location and stimulus, and our touch, pressure, pain, temperature, and taste sensations arrive at that primary sensory cortex we talked about last chapter. The conscious awareness of a sensation is called a perception, while adaptation in this context is a reduction in sensitivity, is a presence of a stimulus that is constantly arriving. So that stimulus is reduced, or the information that's arriving is reduced through adaptation. Now, most sensory information is routed to centers along the spinal cord and brainstem to actually trigger involuntary reflexes. It doesn't get perceived, while only 1% of info from the afferent fibers reaches our conscious awareness and is perceived. Now, our general senses refer to temperature, pain, touch, pressure, vibration, and proprioception, which is our body position. Receptors for these senses occur throughout the body, while our special senses are concentrated within specific structures we call our sense organs that we'll talk about more here shortly. But that's our smell or olfaction, our taste or gustation, vision, balance or equilibrium, and our hearing. We're going to get a little more detailed in Section 2 about our general sensory receptors. They are classified by stimulus that excites them, and we have four main classes. The nociceptors are our pain receptors, thermoreceptors are our temperature, physical distortion from touch, pressure, and body position are stimulated by mechanoreceptors, and then the chemical stimuli or chemoreceptors are our last one. The nociceptors get their name from that prefix nocio, meaning hurt. So again, this is a pain receptor made of free nerve endings common in superficial portions of the skin, joint capsules within the periostea covering bones, and around blood vessel walls. Some deep tissues and visceral organs contain a few. The receptive fields are large, so it's difficult to get an exact source of sensation. They may be sensitive to extreme temperatures, mechanical damage, dissolved chemicals such as those released by our injured cells, and any strong stimuli by any source may excite a nociceptor. Once stimulated, there are two types of axons that can carry that painful sensation. The myelinated fibers carry very localized sensations, are prickling pain, and we call those fast pains. Slower unmyelinated fibers carry these sensations, are burning, aching pains as a slow pain axon. And they're able to identify only a general area in the slow pain type because unlike the fast pain that's more localized. Alright, now there is something called referred pain. This is perception of pain coming from part of the body that are not actually being stimulated. We don't really know the precise mechanism that's responsible, but there are several examples. So in this diagram, it's showing you that sensations originating in visceral organs are perceived as pain in other regions. For example, like your heart. This portion of your body, as well as extending into your arm, is actually going to feel pain, but it's actually a stimulus is actually occurring in the heart. Thermoreceptors have free nerve endings located in the dermis, skeletal muscles, in the liver, and in the hypothalamus. We're going to feel our cold receptors 3 to 4 times more than the warm. They're relayed along the same pathway that carry pain sensations, and they're very active when temperature is changing, but again, that adaptation kind of plays a role here and quickly to stabilize those temperatures. Mechanoreceptors are sensitive to stimuli such as stretching, compression, or twisting. There are 3 classes, and the tactile provides sensations of touch, pressure, and vibrations, while the baroreceptors provide information essential to regulation of autonomic activities by monitoring changes in pressure. And you'll see some overlap with pressure in all 3, but the pressure is of a different context in each one. Our proprioceptors monitor the position of our joints, tension in tendons and ligaments, and the state of muscular contraction. So taking a closer look at our tactical receptors, they range in complexity, fine touch and pressure, while we have crude touch and pressure. The fine touch provides detailed info about the source of stimulation, and the crude touch provides poor localization and little additional information about the stimulus like the fine touch can, as far as location, shape, size, texture, and movement. Now, this diagram here has a lot of info on it. You're welcome to go back in your book and jot down a few more pieces of information, but I'm going to go through each type of tactile receptor real quick and what it is sensitive to. The free nerve endings are sensitive to touch and pressure, and they're between epidermal cells, while we have free nerve endings stimulated by hair displacement in our root hair plexus. We also have tactile discs, also known as Merkel discs. Those are the Merkel cells that make them up, and they have fine touch and pressure receptors in hairless skin. They're close contact with dendrites because located in the stratum basale, and they're going to release chemicals when compressed to stimulate. Our Meissner, or also known as our tactile corpuscle receptors, are sensitive to fine touch, pressure, and low frequency vibrations. You find these abundant in your eyelids, lips, fingertips, nipples, and external genitalia. Our lamellated corpuscle, or our Paxian, are large receptors sensitive to deep pressure and to pulsing vibrations. Common in skin of fingers, breasts, and external genitalia. And finally, our last one, the Ruffini corpuscle, is sensitive to pressure and distortion, but located in the deepest layer of the dermis. And you see a larger picture here that kind of shows the location of them all within respect to each other. Our baroreceptors, they provide essential information to the regulation, again, of the autonomic activities that kind of occurs within our visceral organs. They consist of free nerve endings that branch within elastic tissues, and mostly in the walls of blood vessels, portions of the respiratory, digestive, or urinary tracts. And when pressure changes, the elastic walls of these vessels or tracts expand or recoil. Baroreceptors respond immediately to a change in pressure to try to alleviate that response. And you have a diagram here showing the baroreceptors and regulation of autonomic function. So I would list these, the baroreceptors of the digestive tract, the bladder wall, carotid sinus and aortic sinus, log, and colon, and kind of just briefly jot down those activities. The proprioceptors, they monitor the positions of joints, tension of tendons, ligaments, and the state of muscular contractions. They do not adapt to constant stimulation, and each receptor continuously sends info to the CNS. Most of that info is processed subconsciously, so there are small portions that actually reach our conscious awareness or can be perceived. And your sense of body position results from the integration of info from all 3 of these types of proprioceptors, which are your free nerve endings, your Golgi tendon organs, and your muscle spindles. Now, our chemoreceptors are our last kind here. They detect chemicals. They generally respond only to water-soluble and lipid-soluble substances that are dissolved in the surrounding fluid. Adaptation usually occurs over a few seconds following the stimulation, and their info goes to the brainstem centers that participate in autonomic control of respiratory and cardiovascular functions. They are located in the carotid bodies and the aortic bodies. And again, I have another diagram here for you to kind of jot down the chemoreceptors and what their activities relate to. 9.3 talks about our sense of smell, or our olfaction, and that's provided by our olfactory organs, and these organs are located in the nasal cavity on either side of the nasal septum. Each consists of an olfactory epithelium containing olfactory receptor cells, supporting cells, and stem cells. They have underlying areolar tissue that contains olfactory glands whose secretions absorb water and form pigment and mucus that covers the epithelium. When we inhale, the air swirls within the nasal cavity and carries about 2% of that inhaled air to our organs. Repeatedly sniffing increases our airflow and intensifies receptors. Those receptors are highly modified. Reception occurs as dissolved chemicals interact with odorant binding proteins on the surface of the cilia. The odorants are actually chemicals that stimulate our olfactory receptors. And if you take a look, we have our olfactory epithelium here, the nerves, the olfactory bulb, which we've seen in our dissection, and the tract, and then finally reaching the central nervous system to basically process a smell. The olfactory pathways include the axons leaving the olfactory epithelium that collect into about 20 or more bundles that penetrate the cribriform plate of the ethmoid bone to reach the olfactory bulbs. This is the location of the first synapse. And now these bundles are components of the olfactory nerves. The axons leaving the bulb travel along the tract to react the olfactory cortex of the cerebrum, the hypothalamus, and portions of the limbic system. Because we have extensive limbic and hypothalamic connections, that helps explain the profound emotional and behavioral responses that certain smells can actually produce for us. All right, the last thing we're going to talk about is gustation, which is our sense of taste. And gustatory receptors are distributed over the surface of the tongue, adjacent portions of the pharynx and the larynx. The most important taste receptors are on our tongue. And these taste receptors and specialized epithelial cells actually form our sensory structures we call taste buds. They're well protected from mechanical stress because they're due to chewing. They lie along the sides of the epithelial projections we know as papillae now. The greatest number of taste buds are associated with the large circumvallate papillae that form a V point, pointing towards the base of the tongue, which I'm going to show you a picture here in a second so you can see those here. There's the circumvallate papillae. And this is kind of a zoomed in portion of just one of them you can see here, kind of pointing towards the base of the tongue. The mechanism behind gustatory reception seems to be parallel to that of olfaction. Dissolved chemicals contacting taste hairs, which we know are microvilli. They stimulate change in the membrane potential, which results in an action potential. We have our 4 primary taste sensations: sweet, salty, sour, and bitter. And there are 2 additional tastes actually that have been discovered. Umami, which is a pleasant taste corresponding to the flavor of beef and chicken broth and Parmesan cheese. And then water, which most people say water doesn't have a flavor, but there are actually water receptors present in the pharynx. Lastly, the taste pathways. We have taste buds that are monitored by the facial, glossopharyngeal, and vagus cranial nerves. The sensory fibers of these nerves synapse with the nucleus in the medulla oblongata. Those axons of the postsynaptic neurons synapse in the thalamus. Conscious perception of taste is produced as info is received from our taste buds, and then it's correlated with other sensory data basically to provide our taste. Information from the olfactory receptors plays an overwhelming role in taste perception, and you are several thousand times more likely to be sensitive to taste when your olfactory organs are fully functional. And this is why when you're congested or sick, you may not be able to taste very well because you have a blockage in those olfactory receptors. All right, and we'll pick up next time with the eye, and I'll see you guys next time.